Hey guys, so we're starting a new lesson today, lesson 7.2. It's all about the plasma membrane. So, in this lesson, we're going to be learning why is the plasma membrane an important structure of the cell. Obviously, you guys already know that the plasma membrane surrounds the cell and keeps everything inside the cell, but it actually has a lot more important jobs that it does than just keeping everything inside, right? All right, so here are some vocab words. Phospholipid bilayer, transport proteins, fluid mosaic, selective permeability. You guys probably, I don't know if you've ever heard about any of these, so we will be going through all of them. But then there's the word ion. This one some of you may know. It will come up a few times, even though the lesson isn't 100% about ions. But an ion is any substance that has a charge. So for example, sodium and chloride. Yes, sodium, its symbol is Na, and it has a positive charge and chlorine has a negative. These two are both considered ions because they both have a charge. Chlorine is negative and sodium is positive. This charge gives it special abilities to do certain things, and it also means that the way that it enters and leaves a cell is different than the way most other substances would enter and leave a cell. In case you guys are wondering, sodium chloride is actually just the salt that you eat at home, right? Just salt. All right, so you guys, the first part of the membrane that we're going to be focusing on is the phospholipid. So, if you guys saw earlier here, all of these little things, the little circle with the lines, circle lines, circle lines, those are the phospholipid. They are the major component of the cell membrane. And everything in the cell membrane is basically embedded within the phospholipids. All right, so phospholipids are amazing molecules because they have two very different properties, which gives them the ability to make the whole membrane actually work. So the phospholipid has a phosphate head. The head is made up of a phosphate, and it's hydrophilic. This basically means that the head part of the phospholipid is attracted to water. It loves to be around water. It does not want to be mixed in with oil. You guys know from experience that when you put water and oil together, they don't mix, right? This is because water is not able to actually mix with oil because it has separate properties from the oil. They're not able to interact together. So the same thing with different types of solutes. Some solutes can dissolve in water, which means that they love water, they're hydrophilic, and some solutes can dissolve in oil, which means that they hate water. They're hydrophobic. So the phosphate head, which is this circle right here, is polar, which means that it's hydrophilic. It is water loving. It wants to be around water. And then it's also made up of two fatty acid tails. Yes, that is actually what they're called, fatty acid tails, okay? Fatty acid tails are hydrophobic, which means they are water-fearing. If I bring a fatty acid tail and I try to mix it into water, it will not dissolve in the water. They will basically form a little circle and stay as far away from the water as possible. Just like how if you guys bring a bucket of water and you put one drop of oil, the drop of oil will stay together because they are all trying to stay together away from the water. And only the oil on the outside of that droplet is being exposed to the water. So those are the only ones that are, that are unhappy. So basically, hydrophobic are the little tails. They are water-fearing. Hydrophilic heads love to touch water. So let's just talk a little bit about how these phospholipids are arranged inside of the membrane. So we said the head is hydrophilic, which means it loves water. The tails are hydrophobic, which means that they hate water. The tails are forever going to be trying to avoid water. So in doing so, they form a bilayer. A bilayer basically means that the membrane is made up of two separate layers of phospholipids. So in these two layers, all the tails are facing the inside of the membrane. This means that they're never going to be exposed to the water that is found outside of the cell and inside of the cell, because there's water here and there's also water here, so the tails stay inside the membrane, while the heads are facing the outside where they get to be exposed to the water, because remember, the heads are hydrophilic and the tails are hydrophobic. So this arrangement allows the membrane to keep the internal and external environments separate. So as you can see, the internal environment of the membrane is going to be very different from everything outside and inside the cell. All right, so if you look at the cell overall, obviously this would be a sphere, so it would be closing in on itself, but this is what a bilayer would actually look like. So you've got water outside the cell, and you've got water inside the cell, and the heads are the ones that are exposed to the water, while the tails are the ones on the inside, which are not exposed to the water. So when you look over here, this means that the inside of the membrane is actually an oily environment. Remember how we said water and oil do not mix? This basically means that if I went inside the membrane, not here, but in here, there would be an oily environment in there, which means that certain substances actually cannot enter the membrane. Anything that can mix with water but cannot mix with oil actually will not be able to go inside of this region, which means that they won't be able to enter the cell through normal means, right? So the reason we form a bilayer is so that all the heads are exposed to water on the inside and outside of the cell, and all the hydrophobic tails, remember the heads are hydrophilic, they love water, all the hydrophobic tails, which are afraid of water, will stay together and form an oily environment separated from the water. And they're protected by the heads that are at the top of their bodies, right? All right, so if I ever took a phospholipid and I dumped it into water, they would form a circle like this with the heads facing the water and all the tails facing the inside, forming an oily environment. And if I ever took a bunch of phospholipids and I dumped them into oil, the heads would all face inside together to stay away from the oil, and the tails would go outside so that they can be with the oil. Remember, the tails are afraid of water, but they love to mix with oil. So this is basically what would happen if I dropped phospholipids in oil and in water. Their heads would face the water, their tails would face the oil. Okay? So now, why is this important? Why is it important that the membrane is made up of heads, hydrophilic heads, and hydrophobic tails? So basically what this allows the cell to do is it allows it to control what gets to enter and exit the cell. Anything that is not able to mix with oil actually will not be able to cross this membrane just straight up. So if you look at salt, if you take it and you try to mix it with oil, it's not going to dissolve in the oil because charged particles, like salt, cannot mix with oil. So if this sodium right here, or this chlorine, tried to enter the cell just by squeezing through, it actually wouldn't be able to do that because as soon as it reaches this oily environment, it would be kicked right out. Same thing with large molecules that have some type of charge on them. Glucose has some small negative charges. If they tried to pass through here, this oily environment would immediately reject them and throw them out. This is because they have some type of charge and they cannot mix with oil. So this is actually one of the amazing things that the cell uses to help control what is allowed to come in and what's allowed to leave the cell. While if we look at other molecules like oxygen, carbon dioxide, nitrogen, and water, they have no charges. They have no problems. They can pass straight through this membrane because they're able to mix with water and with oil. If you have no charges and you're relatively small, you'll be able to squeeze through here, mix with the oil, and then come out the other side and continue to mix with the water because you have no problems like that. However, if you were an uncharged molecule and you were large, you actually wouldn't be able to pass through this membrane just because you wouldn't be able to squeeze through all of this area without actually breaking the membrane, right? So you have to remember, small uncharged particles can pass straight in and out of the membrane. So oxygen can come in and water can come in, and carbon dioxide, which is a waste product, can just leave with no problems. While charged particles like salt and sugar and amino acids, or larger particles, they cannot go straight through the membrane, either because they can't mix with the oil or because they're too large. They'll be rejected and they have to find another way to enter the cell. All right, so now guys, that's the end of day 1. Today you have a quiz on Google Forms, so go ahead and give that a try. Good luck, everyone.